Replication accuracy is going to be determined by the shape of the interaction at the active site. So the active site has a very specific shape and it interacts with correctly paired bases, which will have a smaller footprint than incorrectly paired bases. So you can see that an incorrect base pair will have groups that don't fit well into the active site and don't fit with the correct interaction. So a correct GC pair, for example, you see this loop at the top, has an NH bond that can create a hydrogen bond to the enzyme and stabilize the pair in the pocket. Here we have another portion at the bottom where we can have hydrogen bonding to the enzyme. These are stabilizing interaction. We have a polar interaction at the carbonyl group with this top pocket. This is a very specific fit, very specific intermolecular interaction. When you put in an incorrect base pair, these interactions are disrupted. So with this T, you can see you couldn't have a hydrogen bond as you could with a C that is paired to G. And so that doesn't fit in the pocket, and that's going to, because it doesn't fit, it's going to play a role in accuracy. Because if the pair doesn't fit, then it's less likely to result in, then the polymerase is less likely to make this insertion. So if, for example, instead of A, we were putting G in here, then the fit of G would be worse than the fit of A, and the polymerase can reject insertion of G because it doesn't fit correctly. And so mispairs are avoided by the way that they interact in the enzyme pocket. If they don't have the correct shape and interactions, then the then they're less likely to be, then the incoming DNTP is less likely to be sealed to the growing end of the primer. So continuing with replication accuracy. Replication accuracy is improved by, so we said we have some accuracy because of fit of the incoming base pair in the enzyme pocket before it is sealed to the primer. We also have, the polymerase also has what we call an exonuclease activity that is going to proofread the base pair that was just inserted. And this exonuclease activity is most commonly in the 3' to 5' direction. So once the new DNTP has been inserted, then the exonuclease site will read the DNTP that was inserted. If it finds that the wrong DNTP was inserted, it can cut out that wrong DNTP. The enzyme will then reposition back to the polymerase site where insertion of a new base pair occurs to try again to insert the correct base pair. And this exonuclease activity leads to one net error in 10 to the 6th to 10 to the 8th bases added. So it's one error in 200,000 or in 1 error per million or 1 error per 100 million. And then the replication accuracy can be further improved by mismatch repair that comes after replication occurs. There are 5 DNA polymerases in E. coli. DNA polymerase III is the principal replication enzyme. This is the enzyme that is going to load at the replication fork and then process synthesis of replication, so insertion of new DNTPs to the growing daughter strands, and then DNA polymerases 2, 4, and 5 are repair enzymes. DNA Pol1 is going to repair NICs, a missing base pair in the DNA. DNA Pol1 has a variety of roles in bacteria cells. It has both a 5' to 3' and a 3' to 5' exonuclease activity. And DNA Pol1 can be modified experimentally to create a protein fragment that we call Clinal fragment. And Clinal fragment is modified Pol1 in that Clinal fragment is the portion of the enzyme that includes the polymerase activity, and the exonuclease activity has been cut off. So here's an example of Clinal fragment. Clinal fragment, DNA polymerase, holoenzyme has these different portions to it, and it has this 3' to 5', this exonuclease here. If we cut off the exonuclease portion of the enzyme, then what we're left with is this, what we call Clinal fragment. Clinal fragment has the synthesis activity, it has the proofreading activity, but it doesn't have the exonuclease activity, and so the proofreading doesn't really serve a function if we cannot then correct an incorrect pairing. DNA polymerase I is going to perform NIC translation. This is a critical aspect of replication and repair, and we can label probes in the DNA using DNA Pol1. So what's going to happen is that Pol1 will identify a NIC in the DNA. It will remove bases on either side of the NIC. Pol1 will insert new bases to cover that and then seal the NIC. Or it won't seal the NIC. There will no longer be a missing DNTP, but there will not be a phosphodiester seal until DNA ligase is recruited to seal the phosphodiester backbone. DNA Pol3 is a large protein complex. It has many subunits and many functions. You can see that it has all of these different subunits, has several different genes involved in generating this protein, and we have different functions for each subunit. For example, we have polymerization activity, proofreading activity, and exonuclease activity, stabilization activity, template binding activity, loading activity, so how the DNA is then bound to the enzyme for the replication to occur, opening of clamps, closings of clamps, and clamps are what hold the DNA in place while replication is occurring, interaction of single-stranded binding protein with the DNA polymerase. It has another subunit that interacts with the tau and chi subunits. And then we have the DNA clamp itself, which is required for processivity of the template strand as the daughter strand is being synthesized. So the DNA clamp is very important for processivity. There are 3 clamps and we have a clamp loader. So what is going to happen is that the leading strand will be loaded on one clamp, the lagging strand will be loaded on another clamp. The clamp that is loaded onto the leading strand will remain on the leading strand as the DNA is continuously synthesized. And then the lagging strand will have to be loaded each time we encounter a new Okazaki primer. Then the DNA polymerase will be reloaded onto each new primer, and the beta sliding clamp will actually stay with each Okazaki primer, and a new beta clamp will be loaded every time we have a new primer. So this beta sliding clamp can actually stay with the Okazaki fragments, and a new beta clamp can be recruited to bind a new Okazaki fragment.